Over to you, Dr. Rohini.
Yes, good evening, all of you. Am I audible? Okay.
Yes.
Okay. We're going to have group 7 presentation. It was very nice working with this whole group. And here there is again a mixed bag that you're going to have. It's from molecular biology, xenobiotics. So it's again a mixed bag that we are going to have, that is 7, competency number 7. And I will hand over, ask Dr. Ruchir Khare, who was actually the instrumental person to start off with it, will be presenting the slide. So Dr. Ruchir Khare, I hand over the mic. Dr. Ruchir Khare.
Thank you very much, ma'am.
Yeah.
Now I think I am audible.
Yes, you're audible.
Thank you very much, ma'am. And on behalf of Group 7, I, Dr. Ruchir Khare, Associate Professor, Department of Biochemistry, NSCB Medical College, Jabalpur, want to welcome all of you. But before starting the actual presentation, I sincerely want to thank Dr. Sanjay Bedi sir and his entire team, Dr. Sucheta ma'am, Dr. Rohini ma'am, Dr. Nibrati ma'am, Dr. Saiba ma'am, or sorry if I am missing someone, but his entire team has worked very nicely, and all of us are benefited by attending this workshop. So words may fall short if I will start from here only. So I'm not wasting much time. I thank each and every presenter for their crisp presentation and solving our doubts about the CBME. So my special thanks to Dr. Roni Badre ma'am, who was with us since beginning, and when we were not getting anything from where to start, from what to do, she came up and solved each and everything very efficiently. So special thanks to Dr. Roni ma'am from all the team members. And my team consists of Dr. Raj Rajeswari, Dr. Rama Sori, Dr. Reema Mehra, Dr. Rukmani MS, myself, Dr. S. Lakshmi Prabha, Dr. S. Sangeeta, and Dr. Muniraj. They all have put their wholehearted efforts to making this presentation optimum. So I thank my fellow colleague also. So coming to the competency which have been allotted to our group are BI 7.1 to 7.5, and the topic was molecular biology. BI 7.6, and the topic was xenobiotics, and BI 7.7, and the topic was oxidative stress. So objectives of today's presentation are, we have covered along with specific learning objectives for each competency, the teaching learning methods, the estimated teaching hours, the assessment methods, integration part, self-directed learning, and early clinical exposure. Later on, the summary and conclusion will be given by respected Dr. Roni ma'am. So I'm inviting my fellow teammate, Dr. Raj Rajeswari R, to start the SLOs for competency number 7.1 and 7.2. Dr. Raj Rajeswari, over to you, please.
Dr. Rajeshwari, yourself, please unmute yourself. And
Yes,
there are 3 remarks. Which Reema should I unmute after this?
Reema Mehra.
Okay.
Thank you so much, sir. Am I audible to everyone?
Yes.
Yes.
Thank you so much, madam. Good evening, everyone. This is Rajeshwari, Assistant Professor of Biochemistry, Manakalam Nayagar Medical College, Pondicherry. I take this opportunity to thank Dr. Rohini ma'am and all my fellow colleagues for completion of all the competencies and the SDL and EC given to us. And without wasting much of the time, I'll be going into the competency BI 7.1. So BI 7.1, it is to describe the structure and functions of DNA and RNA and outline the cell cycle. Next slide. So at the end of the lecture, the students of first MBBS should be able to describe the structure of DNA with a suitable diagram, enumerate the functions of DNA, describe denaturation and renaturation of DNA, enumerate the various types of RNA, describe the structure and functions of different types of RNA, then explain the phases of cell cycle with a suitable diagram and explain the regulation of cell cycle. So these are the specific learning objectives for BI 7.1. The teaching learning method suggested for this competency will be a didactic lecture, or we can conduct SDL, self-directed learning, or small group discussions. The tutorials can be conducted. Assessment methods, short answer questions and MCQs are the possible questions from these competency, and there is no integration with this competency. 7.1 has no integration. Estimated teaching hour is found to be 1 hour. Next slide, sir. BI 7.2, the next competency, it is describe the processes involved in replication and repair of DNA and the transcription and translation mechanisms. The SLOs for this competency is, has been prepared by Dr. Ruchir Khare, and I'll be presenting the SLOs. So SLO for BI 7.2, at the end of the lecture, the students of first MBBS will be able to define DNA replication, describe Michelson-Stahl experiment to infer the semi-conservative replication of DNA, enumerate and describe the steps of DNA replication, list DNA polymerases in prokaryotes and eukaryotes, compare the replication process in prokaryotic and eukaryotic cells, label the different stages and enzymes involved in DNA replication with the help of a diagram. Next slide, sir. Enumerate or enlist the inhibitors of DNA replication and enzymes inhibited by the inhibitors. List DNA damage processes and describe various DNA repair mechanisms. Enumerate the diseases associated with the DNA repair and type of repair mechanisms involved. Define telomere and telomerase and mention its clinical significance. Define transcription. List or enumerate the types of RNA polymerase and their functions. Enumerate and describe the steps of transcription. Next slide, sir. Enumerate and describe various post-transcriptional processes. Define SNPs. Define spliceosomes. Define ribozyme. Enumerate or list the inhibitors of RNA synthesis and their mode of action. Relate the reverse transcriptase enzyme in HIV. Describe RNA interference and its clinical significance. Define genetic code and describe its salient features. Next. Define translation. Enumerate and describe the steps of translation. Define polysomes. Describe protein targeting and mention the associated disorders. Enumerate various post-translational processes. Enumerate or list the inhibitors of protein synthesis and their mode of action. Compare translation process in prokaryotic and eukaryotic cells. Describe the inheritance of mitochondrial DNA and associated disorders. So for this competency, the teaching learning method are the didactic lecture or small group discussion can be taken, and the possible assessment methods are modified essay question, or we can use long answer questions, short answer question, and MCQs. There is no integration with this competency, and estimated teaching hour was found to be 4 hours. So next competency will be taken up by Dr. Reema Mehra.
Dr. Reema, can you raise your hands? I cannot find any. Okay, it is Reema Kapoor. Okay.
Good evening, everyone. I am Dr. Reema Mehra from GRMC Gwalior, and I am be presenting the SLOs for Competency 7.3. At the end of lecture, the students of first MBBS should be able to explain gene mutation, explain the various types of mutations and their effects, discuss induction and repression of the gene expression, explain the concept of housekeeping and the regulatory genes, enumerate the various covalent modifications occurring in histones which regulate the gene expression, describe the role of motifs in the gene expression, explain the concept of lac operon with the help of label diagram, discuss gene regulation in eukaryotes at the level of DNA, describe how the genes are regulated in eukaryotes at the level of RNA, and describe the gene regulation in eukaryotes at the level of proteins. The teaching learning method for this SLO will be the didactic lectures or the small group discussion, and assessment is via the modified essay questions, long answer questions, short answer questions, MCQs, and the clinical vignette. Integration can be correlated with the anatomy 75.4, the described genetic basis of variations, polymorphism and mutation, or the nesting can be done with the pediatrics department. The estimated teaching hour for the SLO will be are 2 hours. Thank you, ma'am. Now Dr. Rukmini will take the SLO 7.4.
Good evening, everyone. Am I audible?
Yes.
Yes, ma'am.
Yeah. I'll be dealing with the SLOs for competency 7.4. Next slide, please. The competency is the described applications of molecular technologies like recombinant DNA technology, PCR in the diagnosis and treatment of diseases with genetic basis. At the end of this session, students of first MBBS should be able to describe principles of gene cloning with applications of restriction endonucleases and vectors. This is keeping in mind to straight away teach the technique of molecular biology. We have to go with this basic step. Then the next one, enumerate the applications of recombinant DNA, genomics, and complementary DNA library. Describe the basis of gene therapy and antisense therapy, and describe the principles and applications of techniques like PCR, Western and Southern and Northern blotting, DNA fingerprinting, RFLP, VNTR, microarray, DNA hybridization technique, and FISH. So the teaching learning methods is didactic lecture, small group teaching, then tutorials can be the teaching method. Assessment will be short answer questions, MCQ, clinical vignette, and integration is with pediatrics department and general medicine. Both can be of nesting. The estimated teaching hours for this would be 2 hours, keeping in mind all the techniques, principles, applications will be dealt with. Over to Dr. Lakshmi Prabha for the next competency, that is 7.5.
Good evening, everyone. I am Dr. Lakshmi Prabha from Vinaga Missions Medical College, Karaikal. I'll be presenting SLO from Competency 7.5. So at the end of the session, students of first MBBS should be able to, the topic of this competency is describe the role of xenobiotics in disease. Under that, the SLOs are define xenobiotics, describe the phase concepts of detoxification, explain the phase 1 detoxification reaction with suitable examples, describe the phase 2 detoxification reaction with suitable examples, describe in detail various types of conjugation reactions, discuss the biomedical importance of monooxygenase, cytochrome P450 system in detoxification. Next slide, please. Next slide, please. Sir?
Dr. Ruchir Khare, change the slide. Dr. Ruchir Khare, please change the slide. It's come.
Explain the mechanism of detoxification of drugs used in treatment and prevention of diseases. Explain pharmacological significance of detoxification mechanism with suitable examples. The teaching learning method includes didactic lecture, small group discussion, or self-directed learning. And assessment methods include modified essay questions, long answer questions, short answer questions, and MCQs. Integration is not there, and estimated teaching hour is 1 hour. Now I request Dr. Sangeeta to continue the presentation.
Dr. Sangeeta. Dr. Sangeeta. Dr. Sangeeta.
Audible, ma'am?
Yeah, you're audible.
Okay. Good evening, everyone. Myself, Dr. Sangeeta from Government Medical College, Nizamabad. I'll be dealing with the competency 7.6 and 7.7. So competency 7.6 deals with describe the antioxidant defense systems in the body. At the end of the lecture, the first phase MBBS students should be able to enumerate the various types of antioxidants with examples, explain the role of antioxidants in free radical scavenging with a suitable diagram, describe the mechanism of antioxidant defense systems in the body with a diagram, explain the importance of antioxidants in the prevention of oxidative stress with suitable examples. The teaching learning methods related to this competency are didactic lectures, small group teaching, and self-directed learning. Whereas the assessment methods include modified essay questions, short answer essay questions, and multiple choice questions. Integration is nil and the estimated teaching hour is 1 hour. So the competency 7.7, it is describe the role of oxidative stress in pathogenesis of conditions such as cancer, complications of diabetes mellitus, and atherosclerosis. These SLOs were prepared by Dr. Lakshmi Prabha and Dr. Ruchir Khare. So at the end of the session, the first phase MBBS students should be able to define reactive oxygen species, describe the sources of free radicals in the body, explain the mechanism of lipid peroxidation, define oxidative stress, and also to explain the free radical damage causing mutation, cancer, complications of diabetes mellitus, and atherosclerosis. The teaching learning methods includes didactic lecture, small group discussion, and self-directed learning. The assessment methods are modified essay question, short answer question, and multiple choice question. And this competency can be done in integration with the departments of general medicine and pathology. And the estimated teaching hour is 1. So next, the integration part will be dealt by Dr. Rukmini MS. Hand over to Rukmini ma'am.
Hello. Am I audible?
Hello. Yes, ma'am.
Yeah.
Yeah. I thank Dr. Ruchir Khare for preparing this integration part of it. I'm Dr. Rukmini from KMC Mangalore. I could not introduce myself in the last session when I spoke. So this integration, actually these two competencies which we are mentioning here, that is from internal medicine, IM 2.4, and from pathology, PA 32.4, are not listed as integration with biochemistry. But when we found that, which is related to the previous competency Dr. Sangeeta described, explained, with that competency, these two competencies from internal medicine and pathology will be ideal for nesting. So discuss and describe the pathogenesis, natural history, evolution, and complications of atherosclerosis and IHD. So the complications of this atherosclerosis or the oxidative stress, which was in the previous competency, can be integrated here. And nesting with pathology, that is PA 32.4, which says this: classify and describe the epidemiology, etiology, pathogenesis, pathology, clinical laboratory features, complications, and progression of diabetes mellitus. So here we can easily nest or the with pathology department, that is the clinical laboratory features, complications, progression of diabetes, with all the oxidative stress and laboratory parameters related to diabetes. So we have chosen these two. When we look at the competencies in the volume 2, this actually the integration is not shown with biochemistry, but these two are the ones which relate with this competency, that is BI 7.7, describe the role of oxidative stress in the pathogenesis of conditions such as cancer, complications of diabetes mellitus, and atherosclerosis. So I have mentioned this internal medicine competency and pathology competency. So these two which can be used for integration in from biochemistry with medicine and pathology. Thank you. Over to the next presentation by Dr. Rajarajeshwari, self-directed learning. Thank you.
Am I audible, madam?
Yes, yes, yes.
So next I'll be explaining the self-directed learning for this topic, molecular biology. So first we'll be starting with the preparatory phase, and this phase will be with the department. It will be inside the department. So the topic and the topic related questions will be discussed, and it will be finalized by the department faculties, whichever topic we are taking for the self-directed learning. And the finalized topic and the related learning materials like the books or any YouTube links, which it will be informed to the students in the classroom, and also the same will be posted on the institute LMS at least 1 week prior to the self-directed learning activity. And the students also will be informed of the activity, the date and time, everything, and they will be asked to bring their learning materials on the day of SDL activity. So these things will be preparing ahead of the SDL activity. Next slide, sir. So on the day of the SDL activity, the classroom setting, so this first the students, they will be divided into groups, and each group will be given one facilitator, and the groups with each facilitator, they'll be again divided into mini subgroups. Like each subgroup will be containing 5 students. In each subgroups, 5 students will be there, and all the questions what we have prepared in the topic, okay, under the topic, whatever questions we have been prepared, they will be projected in the screen for the students. The finalized questions will be displayed one by one through a projector for the students, and the students will be asked to prepare the answers with a peer discussion. So they will be discussing among themselves with the help of the learning materials they have, and they'll be preparing answers for all those questions. Once the discussion is over, the representative from each subgroup, they'll be invited to discuss the answer for the questions, and all the subgroups will be given equal chances. Following which the faculty will be summarizing the important points. For each question, the faculty can summarize the points. So one example we have taken for SDL activity. So on the topic polymerase chain reaction, we can conduct one SDL activity. So the following are the questions for polymerase chain reaction. First question will be define PCR and mention the 3 steps of PCR. Explain the process of PCR in detail. Mention the types of PCR and discuss the various applications of PCR. So these are the main questions with respect to the first year MBBS students, and they'll be preparing answer for these questions with peer discussion and also with the help of the learning materials they have. So estimated teaching hour for this SDL activity will be 1 hour, and we can assess the students either through short answer questions or MCQ, and we can also use formative assessment during the SDL activity itself. We can assess the students and it can be taken as a formative assessment. So with this, I'll conclude my SDL activity, and next early clinical exposure will be presented by Dr. Ruchir Kare. Over to you, sir.
Thank you very much, ma'am. Early clinical exposure is going to present by myself. I'm myself, Dr. Ruchir Kare. So the topic which have been selected by the group is mutation and mutation detection techniques with special reference to the polymerase chain reaction. The total duration is estimated 3 hours. So the ECE module which is developed, the setup will be classroom or molecular lab or the OPD. Topic of basic science which will be taken is our mutation and genetic testing techniques. So a part of competency number 7.3, BI 7.3 and BI 7.4, which is gene mutation and PCR, was taken. So ECE will be conducted through the case of Fragile X syndrome or molecular testing reports of Southern blot and PCR of Fragile X syndrome by role play or by clinical vignette or photographs. So when I was taking this case of Fragile X syndrome, I was a little confused that whether we are able to see, whether we would be able to see any case of Fragile X syndrome or not. So I have contacted my colleagues in the pediatrics department, and they have said that they have seen few cases of Fragile X syndrome, and in a month or two, there are 1 or 2 cases in the OPD. So you can take this. So with this background, the overall goal which we have taken, the student must be able to recognize the importance of PCR in the diagnosis and treatment of diseases with genetic basis and understand the basic concepts of mutation. The learning objective will be enumerate, the students should be able to enumerate different types of mutations and their effects, describe the basic concept of each mutations, incidence, basic defects, symptoms, and lab findings of Fragile X syndrome, reason out the genetic basis of mental retardation in the patient, enumerate diagnostic modalities of mutation with reference to genetic disease. Students should be able to explain the possible treatment modality in the patient, explain the other different inherited disorder due to mutation, describe importance of Southern blot and PCR in the molecular genetic testing, explain the importance of prenatal genetic testing and counseling to the parents. So the expected competency at the end of the session would be the students would be able to demonstrate the understanding of deviation from normal growth and mental development, correlate the phenotypic deviation with a genetic disease, demonstrate the ability to counsel the parents with genetic defect or mental retardation, demonstrate the optimum attitudinal, ethical, and professional behavior with a patient of mental retardation. In competency number, expected competency number 3, demonstrate the ability to counsel the parents with child of genetic defect or mental retardation. The total time will be 180 minutes and will be divided into small groups, such as for introduction to module, group division and instruction, 30 minutes, exposure to clinical context, means in the molecular lab or cases or discussion of reports, 60 minutes, summary, molecular correlation and conclusions, 30 minutes, Post-test assessment 30 minutes, reflection and feedback 30 minutes. So the case which has been read by the group consists of a 7-year-old boy was brought to a pediatrician by his father with the concern of following complaints. Number 1, boy is having language speech problem, he is hyperactive, he is having mental retardation, which is told by teachers of his school to his father. The father also reported strong family history of mental retardation in the males of the examination. It is found that boy have a large jaw, prominent ears, and enlarged testes, hypotonia, and hyperextensible joints. The examining pediatrician advised the father for few blood tests and to see a genetic counselor, and later genetic counselor stated that his mother, means boy's mother, likely had a silent mutation. The key finding was shown to the student with the help of the photographs, such as long face with large jaws and ears, macroarchidism, hyperextensible joints, and mitral valve polyps in echocardiography. So the teaching methods will be: case scenario was discussed with the students in the class, case discussion with all the available molecular testing reports or clinical case, observation guide. Students were asked to gather and share the information and to observe the different lab results. Within the group, first students were asked to interpret the information they have gathered and to correlate the clinical findings with the lab findings. They were also asked to write down salient observations regarding the case in an orderly manner. The students were also observed for communication and behavioral skill for counseling session through a role play. Facilitator's guide. A few questions which can be put by the facilitator to take the student up to a particular direction, such as how mutation leads to genetic disorder, why the family history is important to diagnose genetic defects, what are the techniques to detect mutations, what are the other disorders which are caused by mutations, what are the important points to keep in mind while dealing with the parents of a child having mental disorder. For program evaluation, after the session, a structured feedback from the student was taken, and a pro forma was prepared by all the peer members and consisting of the questions like: how did you find the content of the session, to rate them on a 5-point scale, such as excellent, very good, good, fair, or poor. And other questions such as what aspect of the session was most useful, which part of the session you didn't find useful, and rate your overall knowledge before and after attending the session. And one open question, any other suggestion for improving the module. Along with this, the reflective writing can also be evaluated. After the session, a feedback from faculty was also taken to know their opinion about the module and to get their inputs for the corrections. So post-assessment was done through a questionnaire which consists of different type of questions such as modified essay questions, short answer questions, MCQs. So we have prepared some sample question for program evaluation, such as a modified essay question: define mutation, 1 mark; describe various type of mutation, 4 marks; enumerate at least 4 genetic diseases caused by mutation, 3 marks; enumerate various techniques available to detect mutation, 2 marks. So some short answer questions such as what is mutation, enumerate techniques to detect mutation with a DNA sequence alteration, enumerate techniques used to detect mutation that affect length of DNA. MCQs with single best response, such as which of the following sentence is not true about Fragile X syndrome. Most common form of inherited mental retardation, FMR1 gene is highly conserved. CGG, trinucleotide repeat expansion of FMR1 gene, and life expectancy is very less. So it is thought that if live patient interaction is not possible, then communication and behavioral learning can also be assessed with simulated patient through role play. Few of the students can be asked to play a role of a parents of a child of mental retardation, and one student can play the role of pediatrician or the counselor. So by this, the cognitive aspect or psychomotor or the affective aspect, all the aspect can be checked or can be evaluated. So this is all about the early clinical exposure, and I would like to request respected Dr. Roni Badre ma'am to summarize, and the session is open for question and answer session after that. Thank you, sir. Thank you, everyone. Over to you, respected Roni ma'am.
Thank you, Group 7. And it was very nice working with Group 7. Everyone was quite enthusiastic. Dr. Rukmini was still today, she was really doing the slides. And Dr. Ruchi and obviously others also have contributed immensely. And it was a mixed bag. So we really had a lot of discussions, and finally we've come up with this. I think the session is open for any more questions to be asked.
Well done, Group 7. This is Sucheta here. And I think, I would like to sort of highlight a few things that this group did a little differently. That program evaluation scale is a good one. It's something that is different. And so well done. If there's anything else that anyone wants to find out or ask, Dr. Bedi, do you have any hands up?
Anyone for discussion? The participation is also very high, good number.
How many people are there? I cannot somehow see the number.
7 are there. Any questions?
Hello.
Hello.
Hello. Good evening, madam. Hello.
Yeah.
Myself, madam. I'm Dr. Suganti. Madam,
Yeah.
can I speak on, ma'am?
Yeah, go ahead.
Thank you, Group 7. We had a very wonderful presentation of the molecular biology plus oxidative stress. Things went very well. Madam, I just have a doubt. Like usually when we start describing the transcription, replication, translation, we teach eukaryotic with a base of a prokaryotic thing. But usually when our university puts on a question and we say that for students, you usually write only on the eukaryotic replication, transcription and translation. Is it what is the same in other universities, madam? Or because there was one SLO written, compare the transcription or translation both in prokaryotes and eukaryotes. How are we going about to teach this, and also in assessment part? Just I want a clarification and just to have a views from other universities also.
Can vary with any universities, because if you look at the competency, it doesn't say eukaryotes or prokaryotes. So when you
Yes,
have
madam.
an
Yes, madam.
differentiate, it will be processed in both prokaryotes and eukaryotic cells. It is university. Each university may decide whether you want only eukaryotes or it could be both prokaryotes or eukaryotes. But according to the competency, it doesn't specify. So the SLO is, I think it is the right SLO. Dr. Dandekar, you
Yes.
can, Sucheta madam, can you comment on it if you want?
I felt that this is fine because you just what you have to do is just show them that chart that we have. Not go into great depth. There is this differences between the DNA polymerases in prokaryotes and eukaryotes. Remember, we give them a table. It's there in
Yes,
my book.
madam. Yes, madam.
Yeah. So So that table can be just shown to them to say to bring out the fact that they're different, because ultimately when we talk, later on, we will talk about the antibiotics and so many other things,
More
and
on
that
that.
time we will be talking about prokaryotes, and then they'll say, "Ye kahan se aaye?" So there should not be a doubt in their heads that they're two different, they are two different mechanisms and machinery that is involved. So I'm okay with that 2.4. I was only having doubts about the Michelson-Stahl experiment. Whether at MBBS level we should actually make them, sort of know it and the tagging and all that. There is no need. They can just know that the replication is semi-conservative. That is my take on it. But it is for others to also opine and say.
Yes. Any more questions? I think Dr.
And
Meghna.
further to that, further to that, when we are talking about the polymerases in prokaryote, and then the next is comparison. Na? So unless you have told them that the polymerases are different,
That's what.
how can you compare the replication process? So either you sort of give them both or don't give them both. So that is the decision that we have to make.
Yeah, that's what I said. The competency says it's a broad competency. It doesn't specify that it has to be prokaryotes. I said university to university.
Yeah. So this will have to be debated at each university level, and I mean it's neither right or wrong. This is that one of the excellent examples of shades of grey.
Ma'am, I just wanted to clarify the same part of it. If a question comes like a structured essay question, it doesn't mention about eukaryotes and prokaryotes. It's generally mentioned. Then a student is supposed to write both prokaryotic and eukaryote, or you just wanted them to write a eukaryotic transcription or translation?
Okay, so at that particular time they would only have to write the eukaryotes, but they will in that introduction, they will say that
Yes,
the
ma'am.
student is aware that there is a difference. That's all. But he doesn't have to go and compare, because the question does not explain compare. So only if he says replication process only is given, but I'm sure the examiner will specify eukaryotic cells.
Thank you,
I
ma'am.
mean, that is where the role of the moderator comes, when the exam paper is set, where a moderator is very essential, and a alert moderator who will look into these small things and see that, that is the reason why I always say, even when I'm setting a paper, you won't believe, even today, after 33 years of teaching, I still ask a close friend of mine to go through that paper. And not only that, I will, as soon as I have set a paper, I will write a model answers to each and every question. So once you write, take a paper to pen and then sort of put it down, you will realize what you have not asked, or where is the mistake when you are beginning to answer. So that is a habit that I think all faculty have to inculcate within themselves. It's not a question of not knowing an answer. Once you have framed a question, write down the salient points. When I say model answer, I don't mean to say write out the whole answer, but at least a broad view. It is a essentiality. Yeah.
Hello.
Hello. Yes.
Hello. Am I audible, ma'am?
Yes, definitely.
Yeah, this is Dr. Sunanda from Hyderabad. I have a query, ma'am. Where in ECE module they have projected regarding the Fragile X syndrome. Suppose that is not available, such kind of the cases are not available in other areas. Then how to go about the? At least we can go for a paper case of any of the mutation related disorder, and we can teach to the students in ECE module.
Yes, of course.
Yeah.
Yeah, sorry. Your call.
Yeah, one suggestion regarding the previous discussion regarding the replication part, ma'am. Many a times we in Telangana, in Hyderabad in Telangana, university question paper many at times reflects regarding the prokaryotic ones rather than the eukaryotic. Could it be replication or transcription or anything? So, but still we are, indirectly telling the students to go for eukaryotic process. But I exactly don't know why the people are insisting more on prokaryotic replication and transcription rather than eukaryotic. Even we
See,
have
one
given
thing I can say
suggestion to
that
our
no,
university
there is no
also.
suggestion, there's no insisting. Suppose because what happened, we all learned knowing the prokaryotic first, and then we
Yes.
said that this is the difference in the eukaryote, right?
Yeah.
So if a student is not really told eukaryote is asked or prokaryote, if he goes about developing the answer, saying that this is the way the prokaryote replication takes place, and these are the differences in the as compared to the prokaryotic, these are the differences in the eukaryotic, I think that is
Yes.
fine, because it all
Okay.
depends on that question that is asked. And if your
Okay.
university feels that okay, that is the mainstay, that because from the prokaryote you can easily just differentiate eukaryotes in a very simple way, that is also
Okay.
fine,
Okay.
but as long as it is labeled properly by the student.
Okay.
That is the
In
main thing.
continuation to the same one,
Okay.
is it that is it the same every university is doing that? Are they providing the model answers to the question paper what they are setting to the university when they set a question paper, ma'am?
Yes, that is the norm for many at many
Yes,
university
ma'am.
levels.
But
And
university is not giving the model answers to the respective colleges to go through that model answer to evaluate the papers many a times.
then that is that is where faculty like us who are enlightened, if I may say so,
Yes,
come
ma'am.
into the
Yes.
picture, where you have to take a
Hmm.
stand now. Time has come where there is no junior, senior, or anything like that. If you
Yeah.
think that there's something, going, I'm not saying this is wrong, but something is amiss, then it is
Yeah.
up to us to go up and say nicely, why should the university, if model answer is there, why should they not give it? They will give it, but you should point it out to them. They are also overburdened with so much work that I don't think
Yeah.
they are in a position to remember. They will have
Okay.
forgotten. It is not janbujke.
Okay. But during our internal exams, we are following the same, ma'am. Whenever we ask our
Very good.
faculty to prepare the question paper, we are at the same time. It is mandate. By default, they have to prepare the model answer. Whatever the points you rightly mentioned, it is not the whole answer word to word, the salient features of the answer. And we circulate among the faculty to evaluate the papers. That is happening at the internal assessment level, but not at the final university examination.
That is where you have to
That
tell
is.
the university people
Oh.
and go talk to the controller of examinations and tell him.
Okay,
Yeah,
ma'am.
really.
Yeah.
Hey, sir,
Okay.
please. Nicely. Don't
Uh,
say I have done
uh.
CBSE course. That is what I'm saying. Just say, sir, please do this.
Okay, ma'am. Thank you. Very nice presentation from this group. Really, they worked very, very hard, and it came so nicely, ma'am. Thank you.
Hello, am I audible?
Yes.
Okay.
Yes.
First of all, congratulations, Team 7. Wonderful presentation and very crisp, very clear. I'm not really going to get into this debate of prokaryotic versus eukaryotic DNA, but I would like to tell you one thing, that a lot of students who come in through the CBSE platform have done a lot of DNA and RNA studies, and this Michelson style experiment, PCR, RT-PCR, they know everything even before they come to MBBS. Apparently, there is a very strong biology curriculum there, which
Yes.
helps
No,
them
it's
to,
true. True.
yeah.
At the
So
10th standard level only.
yeah, 10th and 12th, both. So
9th,
they
10th.
know it forwards, backwards, So what I think is, considering that, of course, we cannot equate to all students, I agree with that. However, as we start talking a little bit about disease, for example, today Fragile X came up in the discussion. I feel we must also be able to talk about eukaryotic, wherever the eukaryotic process is appropriate, we'll have to bring it into the picture, because otherwise, how will students understand the disease? And we cannot say that they will not understand, because the basis for this, and I think even the entrance test, the NEET entrance also, it has quite a hefty portion of molecular biology. So all students have a very firm basis and bedrock of molecular biology in general. So it's something that we can build on, I think. Thank you.
Nice. Any more comments? I think we can call it off, madam.
So if there's nothing, remember to put these up on the CBME so people can once more comment. And yes, point well taken, Pradnya, regarding Messelson-Stahl. It's just that I, felt that maybe it should not be as a part of of the objective. But that is fine. I mean, if everyone else feels it okay, since everyone knows about it, it could be. I'm just afraid that some examiner then should not ask a question on the experiment. It should be in the nice to know area, that's all. Yeah.
Madam,
yes.
hello. Madam, can I just have a suggestion? Can we have an SDL on aging, mutation, DNA repair mechanisms? That will be interesting for students. Aging, mutation and DNA repair. This is a daily part of their life. Then that can be one of the SDL people find very interesting students.
Yes. You know what happens? Most of us, in these groups, we cannot pick on each and every topic of SDL, but the sky is the limit. Each one of us can think of different aspects, and each one of us can give different every year. You don't have to keep repeating the same SDL. So yes, point well taken. Of course, you can have very good idea.
Also, madam, actually, human genome project is not coming anywhere here, but that is one of the interesting thing that attracts people. That was another.
Yes.
I don't know where.
Yeah. Correct. We can put it in one of the. No, Rohini, you think we can? I think yes, that's a good idea. It didn't occur to me. Yeah. Okay. So Ruchir, I think whoever is making those slides can add that and put it into the CBM, into the thing now for discussion. Describe the human.
Yes, ma'am. We will make the necessary correction. I have noted all the points given by the respected members, ma'am.
Yeah.
Ma'am, can I tell you?
Yeah, sure. Dr. Rukmini, go ahead.
Yeah, actually, with all these techniques we have at our institution, we have 3 hours to go about these techniques, principles, applications. Also, as one more faculty just now mentioned about the Human Genome Project, we introduced this also in the end. Since the competencies, we thought of limiting to 2 hours, I did not mention that. Actually, I dealt with that 7.4. In our institution, we give this proteomics, metabolomics, and this introduction to Human Genome Project under these techniques. So we have 3 hours at our disposal to convey this whole thing.
Okay, yeah. Yes. Very nice. So somewhere let it get reflected in your slides.
Okay. Yeah, we'll change that.
Yeah. Thank you. Whoever, I didn't get that girl's name, but that was a good suggestion. Thank you so much.
Yeah.
So should we say good night, Dr. Bedi?
Yeah. Thank you, ma'am.
Thank you. Congratulations, Rohini. Well done. The whole group.
Yeah.
Thank you, ma'am.
Yeah. Bye-bye.
Thank you, respected faculty members. On behalf of Group 7, I say sincere thanks to all of you. Thank you, Sweta ma'am, Nibrati ma'am, Roni ma'am, Sanjay Bedi sir. Thank you, everyone.
Yeah. Good night. Good night. Yeah.
Bye.
Stay safe. See you on Friday.
Thank you, ma'am.